Hi, everyone. My name is Angelina Cheng, and I go to Whitney High School.
I'm Christina Zhao, and I go to St. Marcus Episcopal School.
Hi, I'm Elise Kawai, and I attend Kaimaki Christian School.
And today we're going to be talking about using microbiomes to treat lung cancer. So, within the human microbiome, the majority of the microbial cells reside in the gut. So, because we know there's a correlation between these gut microbes and the overall body's health, we believe that there is a possible cure for lung cancer through the manipulation of these certain gut microbes.
So, what are microbiomes? Microbiome is a community of microbes that inhabit in a particular environment. So, there are many types of microbiomes, which some examples are bacteria, viruses, and eukaryotic microbes. So they're really capable of living in harsh conditions, which they can stand extremely cold or extremely hot conditions.
So there are two main topics we're going to be discussing throughout this presentation. The first one is microbiome, as she mentioned before. So it's defined as a community of microbes that inhabit a particular environment. And the other one is lung cancer, because lung cancer is one of the most hard to diagnose because it's unlike other ones because it's within your body. You can't really tell because it's underneath your skin and within an organ, which is why over 1.69 million deaths worldwide in 2015 occurred. And so far in the United States alone, this year, over 234,000 people died. And our topic we're trying to answer today is, by manipulating specific microbes in the gut, are development in the lung cancer treatment possible?
Depending on its success rate, the proposed treatment may reduce or provide an alternative to traditional radiation and chemotherapy treatments, both of which share severe, sometimes permanent effects. These side effects can include fatigue, nerve damage, increased chance of infection, and tissue damage. However, since some microbiomes can be affected by targeted medicine, as we learned this week, this form of treatment may have less harmful side effects. Consequently, there will be a higher quality of life for patients in addition to longer lives. Moreover, should this form of treatment work, a similar approach may be possible with other cancers. In November 2016, University of Texas, Dr. Jennifer Wargo studied over 100 fecal samples from people with advanced skin cancer. She ultimately discovered that patients with more diverse types of bacteria in their gut responded better to immunotherapy treatments, thus supporting the hypothesis that gut microbes have a noticeable effect on health. This is backed by another experiment in which microbial-treated mice who were injected with liver cancer had 40% smaller, as well as less heavy tumors than their untreated brethren. Moreover, nanoscience is becoming increasingly integrated in medicine. This year alone, there are currently over 80 active or recruiting clinical trials for cancer nanotechnologies. In order to develop a means to fight and perhaps even cure lung cancer through the manipulation of gut microbes, we will need to research first, as relatively little is known about these microbes. Similar to Dr. Margo's approach, fecal samples can be collected from patients with lung cancer and analyzed with confocal microscopes, a form of nanoscale microscopy. If more resolution is needed for our samples, we are considering using atomic force microscopes, since it also does not damage biological samples. After analyzing the data pool, we can determine which microbes respectively help the body resist or succumb to lung cancer. Two separate medicines can then possibly be developed to stimulate the former and to kill the latter. We could potentially use bacterial contractile nanotubes, which are highly precise in our cure. Additionally, this process might give us more understanding to how the gut microbiomes affect health and lead to the identification of more microbes.
So some of the challenges we might be meeting is that to create the the data to extract the data from the microscopes and stuff, it takes a very lengthy time to analyze the data also, and to be able to have the confocal and the atomic force microscopes. It costs a lot of money, so it's kind of it just costs a lot. And so as living humans are part of this study, there's potential for other factors to corrupt the data. So due to some facts that are really we don't really know a lot about it, so some inferences and interpretations will be made. And so for our proposal, one would be we get to study the microbes in relationship to lung cancer, and two, from the data gathered, we will develop a new treatment for lung cancer. So basically, the second goal might not be reached, but if it do happen to get reached, it could be a new treatment for lung cancer, or it could work with some kind of medicine that already exists today. So work together to cure lung cancer. And so further information can be researched and used for future studies and research.
Angelina and Christina jointly developed slides 3 and 8. Angelina edited slides 1, 2, 4, and 10, while Christina edited slides 1 through 4 and 8 to 10. I came up with the proposal idea and did the development research for that, as well as created and edited all the slides except for slides 3 and 8. Christina and Angelina jointly used 3 out of the 19 entries on the works cited list, and I used the remaining 16. This is our works cited list. Thank you for coming to our presentation, and thank you for listening.
So do you guys have any questions about anything?
Can you guys clarify a little bit more about what information you guys would want to gain from your system by using atomic force microscopy with the two cell types, I guess your lung cancer and the microbiome cells? What information are you trying to access with the technique of atomic force microscopy?
We were hoping to access exactly what microbes do affect, or perhaps do affect lung cancer, whether they help the body resist lung cancer, or whether they accelerate the effects of lung cancers.
Any other questions?
I have a question. So in trying to identify different parts, positive contributors versus negative contributors in the microbiome for prognosis with lung cancer, how would you try to differentiate or identify them?
I think that we would try to differentiate them and identify them by also keeping aware the prognosis of the individuals from which we are asking for samples. In Dr. Wargo's experiment, from which we were inspired for this specific or our own idea, she kept track of the prognosis, and then using the samples, the people who all had more diverse bacteria, for example, were the healthier, they responded better to treatment, while the people without, so we would be looking for trends.
How did she measure healthy bacteria versus not? Just total amount? Or?
I did not have access to her full study at the time, although I did check to make sure that the source that I found her results was reliable.
Any other questions?
So you mentioned that the experiments that you would be doing are somewhat controversial. So for the people who are kind of like, why are you doing this? This is not a good thing. What would you say about the negatives? Like, why do you think that
I realize that while it can be time-consuming, I do think that nanoscale microscopy has a lot to offer in the realm of medicine, and that there are in fact already drugs that have been developed from nanoscale science. So I do think that also we talked about at a point in science where there are certain limits to every single approach, but I think that in addressing certain limits that we have now in medicine, that nanoscience can help fill in a lot of the gaps.